Thank you all for being here, and welcome to the first biannual NITRO OA Model Symposium. I'm, for those of you who don't know, Dr. Ross Urick, the Program Manager of NITRO at ARPA-H, and I sit in the Health Science Futures Mission Office. Without further ado, let's get rolling. So let's start with what NITRO is. And if you scan this QR code behind my head, that will take you to the NITRO team page or the NITRO website. You'll be able to learn more about what's going on through the course of the program and also learn what NITRO is. When we first launched NITRO, it was the first program from the agency and was the first program from Health Science Futures, and it sought to eradicate osteoarthritis through targeted regenerative therapeutics that will revolutionize the care algorithm, prevent pain, decrease the economic burden, and eliminate the need for repeat joint surgery with equitable access for all Americans. We do that at ARPA-H with technical areas, or TAs, and we have 3 TAs that are the primary focus of the program. And I'm saying this to then say why we're here. TA1 is needle-based and/or non-invasive subchondral bone regeneratives. TA2, needle-based and/or non-invasive cartilage regeneratives. And the real vision here is, let's say you come in with just cartilage degeneration in your knee. It's at a clinician's discretion to be able to say you need bone, you need cartilage, and put something in your knee, one and done, you never need a procedure again. For those patients who need a reconstruction, need open joint surgery, and have no other recourse, there's TA3. Allogeneic and autogenous non-immunogenic, load-bearing, and osteochondroinductive total joint replacements focusing on the knee. And in the ARPA-H fashion, we do things a bit more aggressively, kind of like the DARPA model. So for the program, there will be 24 months dedicated to therapeutic discovery. That will consist of in vitro and small animal model studies. Then we transition to preclinical, and that is really why we're here. Preclinical is 18 months dedicated to large animal studies. Now, in the course of building this program and doing a landscape analysis, we spoke with a litany of people who are the key stakeholders across industry, academia, regulatory partners, the federal government, and everybody has their own answer for what the best large animal model is, which means there is no answer for the best large animal model and why we're here. So in this program, and what we aim to identify with the symposium is that ideal large animal model to better inform the industry and to better inform the program, because our performers will then use the consensus large animal model that we determine here in this symposium today and in future symposia. They'll commit to that, publish on it, and then use it in the preclinical model to streamline and equilibrate and hopefully get to the regulatory endpoints that we need. If they're able to show that they're meeting all the NITRO metrics critically in the preclinical model, they then progress to phase 1 clinical trials. When we launched NITRO, Secretary Becerra, who sits right across the street at HHS, was kind enough to say that the era of ARPA-H is here. NITRO will revolutionize the entire field of regenerative medicine. That's what we aim to do. But it's not just the science. The science is great, though. We aim to eradicate the disease, reversing damage, eliminating the reliance on replacements, and if a replacement is needed, no revisions are necessary. But as I've said repeatedly before, great science is not just the answer here. We're not here to just advance basic science, and if we create $1 million therapeutic, we fail as well. We're here to create deliberate, efficient, and effective forward progress through a variety of means and methods. One of them is this symposium. Additionally, OA clinical trial stakeholders, FDA involvement, and you'll hear from the FDA today. PATIO team, so PATIO is our internal public market accelerator to get things to market. And success points for us will mean that there is a clear path to commercialization and market, not just an advancement of the basic science industry. And integrated equity metrics. So ARPA-H's mission is to meet Americans everywhere. What that means is that for osteoarthritis, in the context of NITRO, the disease is not the wealthy golfer disease. It's in fact quite the opposite, and we'll talk about that in a moment. So each performer team had to come to us with an equity officer. We then approved that equity officer, so an EO per team. We will also have a fractional equity executive running the team with us, who will make sure that these checks and balances are met to ensure that we have appropriate KPIs, that we can build the roadmap to equity, that we can have an insurance action plan accounting for the Indian Health Service and DHA, CMS, TRICARE, and we make sure that we account for every possible demographic to meet Americans everywhere. But why are we here? Museum of the American Indian. As you can see on the bottom right, OA is the number 3 cause of disability. Currently affects 32.5 million Americans, and in the next decade, it's projected to be the number 1 cause of disability. In that population, so those 32.5 million Americans, 20% are Native American and Native Alaskan. It's the highest for multiracial non-Hispanic and Native American and Native Alaskans, and it's 2 times more common in females. OA will continue to erode active years and the quality of life in greater proportions for the American Indian. And this represents the first of many engagements with the American Indian community. Future engagements for us and from our team will consist of listening sessions with IHS, patients, providers, and caregivers to account for what their healthcare experience is like and to guide our performer team. So an example of that is, let's say somebody only takes a bus to their doctor once a year because they live on a reservation. That has to be accounted for in what therapeutics we create downstream. We're going to also require preparation of an equity map, a roadmap to equity, creation of an insurance action plan, generation of those KPIs I mentioned that directly account for the American Indian patient, enrollment of Native Americans in the NITRO clinical trials, and active coordination with a plethora of tribes, accounting for the fact that somebody in Oklahoma and Alaska will have very different experiences with the healthcare system. The vision for this symposium is fairly simple, we think. We aim to bring together diverse communities of experts in regenerative medicine and OA to identify the ideal large animal, primarily, and the in vitro models of OA. This, in turn, will streamline the translational pipeline, ultimately decreasing the price of therapeutics for all patients. While I mentioned that there's an insurance action plan, the part that's underneath that is people don't have insurance sometimes, and we need to account for that in the pricing models for what we create. You can register on our website, as you did before, and we'll keep you updated. Check our website. You're also welcome to email us at any point. The target audience is you, but you probably don't know who all the other people in this room are. So you are the OA and regenerative medicine experts from academia, industry, minority-serving institutions, veterinarians, contract research organizations, manufacturers, federal stakeholders, and regulatory experts. We brought the best and the brightest here, and we also acknowledge that by virtue of this being in person, there is inherent bias built into this. So the next symposium will be entirely virtual to account for that bias. The results that we want at the end of this should be able to be equilibrated across the landscape for OA and regenerative medicine. Today's focus will be fourfold. It's a snapshot of cutting-edge large animal and in vitro OA research. It is not comprehensive of what's going on across the federal government, but it's a very good assessment of what we're doing and what we're looking at. Regulatory considerations for OA models. We'll start the initial conversations to establish the ideal models, and we ensure that those ideal animal models will account for critical OA demographics and ensure the therapeutic access and equity for all Americans. I mentioned this is biannual, roughly February of next year. We'll start talking about the next one, or we'll announce that, and that will be, again, totally virtual. This will end Q4, so federal quarters and federal years are a bit different, so our fiscal year starts October 1st, but Q4 of FY25, so that by the time we hit preclinical, all of our performer teams, we call the performers, not grantees, all of our performer teams have time to adjust and course correct to that consensus animal model. So you filled out a survey when you registered for this, and as you are able to see here, and you'll be able to see on the next slide, everyone has different experience with the FDA. Everyone has a different experience with an animal model. That's also why we're here. 62% of you have never engaged with the FDA. Most of you have only engaged in preclinical research. And of those of you who have engaged in the IND capacity, only 19% of you have ever used large animal models. Of those who have used large animal models, and this is the best part, you're all evenly split. Pretty much down the line. Dog, sheep, horse, pig. Some of you chose non-human primates, and then the rest, very generous. Goat, not applicable, no preference, just here to learn. The induction method of OA was, again, pretty evenly split, but primarily DMM, whether it's open surgical or arthroscopic. For small animals, it's a little more consistent with what we would expect. So rat, mouse, and mostly ACLT and DMM. Structure for today, you're obviously hearing from me. You're then going to hear some phenomenal speeches from NIAMS, NCATS, the FDA, NASA, the International Space Station. Then I'll close out, we'll go get some lunch. We'll come back and actually discuss and start picking your brains about what the best approach is moving forward. You have these prompts, but just so you know that there are 7 topics. We're going to rotate. We'll have notepads. There will be a recorder at each table documenting on your behalf. And then we'll then have a post survey. You've got the QR code at the end of the day, so you got to stick around for that one. Those QR codes and those surveys are critical. So the pre-survey and the post-survey is really how we reflect what you're thinking, what you're learning, how we're growing, how we're adapting, and will help guide future symposia. So if at the end of today, everyone says, we're using the horse, okay, that would be great to know if we're going to use a horse or a dog, but it's going to help us guide future symposia to see where we should be, collaboration with our regulatory partners, collaboration with our federal partners and stakeholders and the experts in the space to know if we're going in the right direction here. So please do the surveys. And again, I'm Dr. Ross Urich. It's an honor to be with you. It's an honor to be in this museum. That's the QR code for the program. And without further ado, I'd like to introduce Dr. Gail Lester from NIAMS.
Hello, and thank you all for being here. I took the pathway rather than the steps because I have 2 hip replacements myself. So it's without a railing, it's better to take a walk than a step. So thank you all for being here. This is a really exciting opportunity. I've been working at the National Institutes of Health since 2001, and I have been, my research has been focused on clinical osteoarthritis. So I'm not doing research, but my research area that I've been supporting. I'd like to thank Ross and the team here for inviting me to come and represent the Institute. I also have one of my colleague program directors, Dr. Ted Tsang, who's here with me today. So the 2 of us can help if there are any questions related to my presentation or to the Institute's focus in osteoarthritis. Today, we're going to be looking more at models, and I'm going to just start out by saying that although as titled, these are comments and not recommendations that I'm putting forward. So I'm just going to be giving you a little bit of information about what the Institute has supported with regard to research with animal models. And I'm going to be using some slides from some work done by Dr. Brendan Lee and his teams that he's collaborated with that use the large animal models for his research. It doesn't mean that that's the only group doing this. It's just that that was a way to show you some of the capabilities of the particular model I'm going to be talking about. So let's see. Okay. So there, again, my first statement, NIAMS has no recommendations or guidelines for specific animal models to be used. Preclinical models, mouse or rat, as Ross has mentioned to you, are frequently used for safety and efficacy. And then there are numerous models that are employed by investigators doing research on cartilage repair and regeneration, or ACL ruptures and injuries associated with that. We do not take a stand on which models should be used. This is decided through the peer review process when grants are submitted to our institute. And we equally fund research in a number of different animal models, depending on the value or the evaluation of the study section review. Now, FDA does have regulatory requirements, and I'll talk about that just a little bit, and we have a presenter from FDA. We'll cover that in much more depth. But here are just a few of the models, and Ross had them in his slide. I think we have the mid-sized models, the dog, the pig, and then the larger size, the cow and the goat. These are, the cow and the goat are a little bit less frequently used. In the larger animals, and that's what Ross had asked me to speak to, the equine model seems to be the most widely used amongst the investigators that we are funding in this area of osteoarthritis. Now, if you're talking about ACL injury and post-traumatic OA, that is a completely different area, and I'm not addressing that today. So part of the reason that the horse the equine model has been widely used is that horses naturally develop OA in a manner similar to humans, and they do share some similarities with joint mechanics. These are some slides from Dr. Alan Nixon and Brendan Lee's work, were published a number of years ago, but they show you that this is the horse hawk model, and it you do a burr. Let's see, there's a pointer here. Maybe here. Oh, yeah. So a burr injury, and then over time, and these have some treatments here, but down here is the no treatment sham. the sham, no change, but the no treatment PBS, you can see this significant injury and then the changes that occur over time and are prevented somewhat by some treatments that have been used. So this staining in here is showing some of the injury. Now, the next slide shows it a little bit more in depth. And here we have the saffron O staining of the osteochondral chip model. And you can see this is normal. And then with the injury, you can see the the loss of staining for saffron and O. And then with some treatments, we can reduce this and maintain the saffron and O, which represents proteoglycan staining. So this is just showing the capability of standardizing injury and being able to possibly see effects of treatments. And that's what we're all looking for in these models. This is another example, and this is one where you can see that you can quantify the changes that occur with the injury. So this is no treatment, and the no treatment has significant loss of cartilage volume and cartilage surface area. And then with these different treatments, and then combine the two treatments, you can see that you're preserving the cartilage volume and the surface area. So finding a model where you can quantify the damage and be able to see the impact of treatments is very, very important for these models. And I think that's part of the reason that the horse model has been pretty widely used in the community, in the research community, and seems to be pretty reliable. Now, we're not endorsing this model as such, because any model, if it does show the similar characteristics, this is purely an example of what a model could do for you and how it could help you understand whether the treatment you're proposing to use, be it some kind of drug treatment or a treatment with a vector. I think in some of these cases they've been using IL-1 receptor agonist and antagonist to try to alter the inflammatory state in the joint. So those are that's everything I want to say on animal models right now. And the take-home message, which I've already stated, that there are a lot of opportunities in the literature showing you different animal models. A lot of we've published, investigators have published many papers that show the weaknesses and strengths of all these different models. There are also regulatory guidelines in the literature that can help you with identifying the best model for the particular purpose that you're trying to serve. One of the things that I think is important is that the investigators try numerous models to show the efficacy of whatever treatment they're proposing. So I think it's not an easy answer to say what the best model is, and that's why we brought everyone together here, is to try to get input from as many voices as we can hear and try to help Dr. Ulrich find the right model as they go forward with this very exciting and innovative project. So the second part of what I'm presenting to you is just kind of a brief introduction to in vitro systems and organoid models. And this is going to be covered much more fully by the next speaker. And we at NIAMS do support research in this area. And these three-dimensional systems are really quite detail. There's tissue on a chip, and then there are organoids. And both of these are very rapidly becoming the go-to systems for understanding the impact of treatments on cartilage and other organs. You can put together a tissue on a chip that has tissues from liver, from the joint tissues, from synovium, from cartilage. And those experiments with those tissue on a chip, and I think that's going to be covered more in the next talk, are really informative and can be very beneficial in figuring out what the next steps need to be in your process. So I put some points here that these methods can be helpful in figuring out how to move forward and what next steps would be. And there is a big movement, and the next speaker from NCATS is going to emphasize this a little bit, to move away. And most of you are probably familiar that Congress has been trying to encourage us to move away from animal models. And what's been developed in the last probably 5 to 10 years has been very informative and helpful with us moving in those directions. So one more point to make is that and that's the last bullet that wait a minute, can I go back? Maybe, yeah. The last bullet here, the morphogenetic processes in these organoids, and this is kind of, I think it's maybe either right behind or right at a similar level to the tissue on a chip, that they may differ from those in growth and development. So there's a lot of complexity in the organoid models that I think is just being really explored at present. So we do have well, did I bring that example? No, I didn't. But there has been some very sophisticated work done with the tissue on a chip by Doctors Lin and Goodman. This is some that was supported by our institute. And they were quite, the paper I would refer you to, and we can probably make this available to you, but it was published recently, is very, very promising because there seems to be a very robust a matter of robustness to this model that can enable you to study both the joint pathology and the treatment efficacy of different therapeutic interventions. So those models are really works in progress and are actively being pursued by a number of different groups. So my purpose here is not to endorse any particular one, but to give you some of the most up-to-date information that we have. And that's all I'm going to present. And I look forward to this afternoon of really robust discussions and more input, and would be delighted to answer any questions. I don't think we have questions now, but maybe at later part, Ross, is that right? Later? Okay. So thank you very much for your attention.
So good morning. My name is Pasley Hargrove Grimes. I'm a program officer for the NIH Tissue Chip Program. And I'm on behalf of me and my team at the Office of Special Initiatives at NCATS. I want to thank Ross and the other organizers for the opportunity to be here with you today and to give kind of a overview of how we are developing in vitro tools, tissue chips for osteoarthritis drug development and precision medicine. So the National Center for Advancing Translational Sciences was started in 2012 with the goal or the mission of turning basic research observations into innovative and dynamic health solutions using the principles of translational science. Our goal here our vision is very simple. We want to bring more treatments to all patients or all people more quickly. We are completely disease agnostic. We study rare and common diseases. We study Mendelian, non-Mendelian, sporadic diseases. Really, if there is a disease, we're interested in it. So I did say I work for the Office of Special Initiatives under my incredible mentor, Dr. Danilo Tagle, who couldn't be here today, which is why I'm here with you. I'm excited anyway. And also our Deputy Director, Geeta Senthil. And really our mission is to address translational problems using innovative solutions through the development and implementation of disruptive technologies that could potentially result, and we hope it will result, in a paradigm shift within the field. This is really an overview of all the different initiatives that kind of span our office. And we put this slide together to kind of show you where in terms of where they're falling in terms of fundamental research all the way to regulatory acceptance. There's a key with the colored key on the bottom to show you what type of technologies I'm referring to. Today, I'm going to be talking to you about tissue chips or microphysiological systems for drug screening program. This program began as a flagship program at NCATS in 2012, and so we're very happy that it's still going after 11 years. We developed the technology, and then we moved into demonstration. We've really shown robustness, reproducibility, reliability, and relevance of tissue chips, and we're now in the dissemination portion, where we're really trying to work with the FDA for eventual regulatory acceptance of some of these tissue chips as bona fide drug development tools. So the public health challenge. As you know, the time to develop a drug takes extremely long amounts of time, so 10 years and almost $2.6 billion to bring a drug to market, and that actually includes the cost of failures. So despite significant improvements that we've seen in vitro biology and toxicology, as well as computational biology in the past 2 decades, we still know that only about 5 to 7% of human diseases have available treatments, and that this value is even lower for patients that are struggling with rare diseases, neurological diseases, or diseases that affect vulnerable populations, such as pregnant individuals or pediatric populations. And so with this, I can say that 9 out of 10 promising therapeutic candidates that enter clinical trials fail. 55% are failing due to lack of efficacy, so they simply do not help us ameliorate disease condition. And then 28% fail due to unexplained toxicity that was never shown in animal models or the 2D cell culture models that were used to file this IND. And so we can surmise from the figures that I'm giving you here that we need to find alternative models that could potentially help us improve the drug development pathway. So I like to do this slide. This is a quote from George Box. He's a British statistician from 1976, and he said all models are actually wrong, but some are quite useful. I actually tend to agree with that. There's a famous historian that many of you have read his book, Sapiens, Yuval Noah Harari, and he said, scientists generally agree that no theory is 100% correct. Thus, the real test of knowledge is not truth, but utility. Science gives us power. The more useful that power, the better the science. So what I'm trying to show in terms of the models at the bottom, and this comes from the paper from Hang Lee's group that was just published in Trends in Biotechnology in 2023, so this past year, showing that really models are a trade-off between throughput and complexity. There is no model, I believe, that is going to answer every single pathological question, but there are questions that we can answer, and that's what I really want to focus on. Tissue chips here that I'm going to talk to you about today really sit between explants, which are, either bioprinted tissue or just, pieces of tissue, and also animal models. We know that animal models are the most complex in terms of their physiology, but we also know that there's absolutely no throughput with animals, really, versus 2D cell culture systems that have massive throughput that can be used for drug screening, but have very little complexity and may not be very pathologically relevant. So this trade-off is important, and it's something I think that as scientists we all have to think about. I'm talking to you today about tissue chips. You've probably seen these described as organs on chips or microphysiological systems. We have not yet as a community agreed upon the terminology or the standardization for this terminology. The FDA has created what I think is some of the best terminology in terms of what tissue chips are, what organoids are, et cetera, and you can look that up online, but that still has not been universally agreed upon, and it's something that we're going to have to work together as a community. Tissue chip technology for us is more physiologically relevant than conventional models for the reasons that I list here on the screen. Oh no, I wanted to go back, sorry. So we can precisely control the fluidic environment. We can provide oxygen and spatiotemporal chemical gradients. We can provide nutrition, and we can remove waste products. We can create these dynamic biomechanical cues. And we have integrated inline sensors to monitor tissue viability and function. These inline sensors are extremely critical because most of these experiments involve taking an actual tissue chip and at the end kind of killing it off for endpoint analyses, and we would like to prevent that if we can. And recently, to make these more physiologically relevant, we've added features of the circulating immune system, as well as features of the microbiome. So what have we been up to for the last 11 years? We have actually been focused on three main areas. We started in 2012 with really low-hanging fruit, asking ourselves, can tissue chips predict safety and toxicity of compounds that were shown to not be toxic in animals, but then were later found to be toxic in humans. And we were able to actually do this. Our teams published over 500 peer-reviewed papers, many of them in top-tier journals, showing, in fact, that tissue chips are very predictive of toxicity. 5 years later, we moved into the disease modeling and efficacy testing space. As I said, we are disease agnostic, so we are looking at all different diseases, and we have a number of initiatives that have fit into this. Our Tissue Chips for Disease Modeling and Efficacy Testing program is what I'm going to talk to you about today, but we've also worked extensively with NASA and ISSNL to send tissue chips up in space, and they'll be talking to you about that today. We've also worked with NIDDK to model type 2 diabetes on chip, as well as NIBIB to model functional immune system aspects on chip. And lastly, we've also worked with the HEAL initiative, or helping to end addiction long-term, to use tissue chips to model nociception, addiction, and overdose, and perhaps find non-opioid analgesics that can be used in the future. Within the last 3 years, we've tried to push into the precision medicine space, asking ourselves a very simple question. Do tissue chips have clinical utility? Could they eventually be used by clinicians or be used for clinical purposes? And so to do that, we've started our clinical trials on the CHIP initiative, asking if tissue chips could be used to inform the planning, execution, and design of clinical trials. Today, I'm really excited to talk to you about a program that just ended, Tissue Chips for Disease Modeling and Efficacy Testing. The goal here was to really develop models of human disease using tissue chip technology to test efficaciousness of drugs. So to also assess biomarkers, to look at bioavailability, and also on and off-target effects of target engagement of candidate therapeutics before they enter into clinical trials. So this program really requires, and I like to stress, the interdisciplinary expertise that this program requires. We have experts in most of these projects. We have scientists working alongside clinicians, people who are skilled in material science, omics, chemists, electrophysiologists, pharmacologists, you name it. So we really try to bring really diverse teams that can work together to answer these types of complex questions. And not to belabor the point, but we are here talking about osteoarthritis. You know that there's a huge unmet clinical need for OA. This is the most common joint disease, as Ross said, affecting over 32 million people in the United States and being the leading cause of disability in older adults. The risk factors are both controllable and not controllable, right? If any of you can come up with how we can fix aging, that would be excellent. I don't think any of us, I think we're all tired of aging, so I understand. But then mechanical overloading and obesity. The obesity in our country has increased dramatically. There are lots of reasons behind that, but that is a major risk factor as well. You can see within the disease on the right that you can really see structural changes, including cartilage destruction and the narrowing and loss of this joint space, leading to a lot of pain, stiffness, physical disability, and fatigue, not to mention the emotional fatigue of being chronically in pain and unable to do anything about it. So there are no efficient disease-modifying drugs, and that's what we need to work for. So the project I'm going to talk to you today comes from Hang Lin, Bruce Spinell, and Stuart Goodman. And this really just wrapped up recently because of COVID delays. The project was really extended about a year to really finish up milestones and key aspects of the project. But this is a human cell-based microphysiological and multi-component joint tissue chip used for drug testing. You can see that there are four main components: adipose, synovium, bone, and cartilage. You have inlets and outlets on the top for the specific media, so adipogenic media, fibrogenic media, osteogenic, as well as simulated synovial fluid that's able to flow through the bottom. And so I actually think, and I'm going to try to show you the recent video that was published. I don't know if I can click it. Let's see. I may just be out of luck. So the team actually came up with, they were selected for a Joe video to show you how you put the chip together and then the main results that they see with the chip itself. But just to kind of, since I can't show you the video, I will go over kind of the major accomplishments from the team. They were able to use mesenchymal stem cells to create the knee joint or miniature knee joint on a chip. This allows novel communication while maintaining the respective mature tissue phenotypes for around 8 weeks. When exposed to IL-1 beta, you can see IL-1 beta mediated inflammation similar to what you would see in the clinic. You can also add drug treatments to look at therapeutic effect of different disease-modifying drugs, which they did with naproxen, and they were able to see some effects in terms of reversal of OA phenotype. And the introduction of drugs into different tissue media, either individually in the top or collectively in the bottom, really allows us to simulate in vitro systemic administration versus intra-articular administration, which we would also note clinically. So we have a lot of I didn't mention, and I wanted to, that in addition to naproxen, the team has also been looking at novel interventions as well, so exosomes, as well as decoy oligonucleotides that potentially could target the NF-kappa B pathway. But in addition to drug testing, this in vitro system also has a lot of potential advantages for precision medicine. So in this graph, which was from their most recent paper, and I think is very, very well done. You can see that you can take in panel A, you can take arthritis patients, and you could do biomarker analysis to basically stratify them into unique patient subsets. And then you could actually model the unique pathological changes that are occurring in these patients, either on chip or in animal models. And you could do some drug screening as well, which you could see here from 1 to 6. And to make this truly personalized, you could take somatic cells from a patient and you could reprogram them into iPSCs and then further differentiate them into the cell types of your choice to essentially create a personalized you on a chip or a joint chip that's personalized to you. If you add EHR data, electronic health records, as well as data from genomic analysis, pharmacogenomic analyses, you can essentially have a really complete picture of what was happening with a patient, which would give you the opportunity to really test a multitude of drugs. You wouldn't end up affecting the patient, but that would not end up hurting the patient. You would be able to find the ideal optimal drug. And they put this slide in as I put this slide in as well from their paper to really demonstrate that the cross-validation of in vitro and in vivo models is extremely important. I do think it's going to be impossible to recreate every facet or component of a disease pathology within either a joint chip or within an animal model. And so being able to actually use both of these models and as comparators really, I think is critical as we proceed more into the future. And this is just showing you that you can collect the tissues at the end in panel B and perform online, inline, and offline analyses to really look at kind of downstream processes that are occurring. So there are a lot of challenges that are impeding uptake of tissue chip technology, and that would include the osteoarthritis chip or any chip that you can think of. The first is that we don't have standardization of these devices, and we have not yet agreed upon how to standardize them, and that is going to be a long work in progress. We did not see really the advent of tissue chips until 2010, and so it's only been about 13 years, so relatively new technology. So we are going to have to wait for that. We also need detailed use cases for tissue chips and how they can replace or support existing drug discovery assays. It would be really great to benchmark tissue chips against the current gold standard assay to be able to actually make sense of if tissue chips, how valuable they are. There's an extremely high cost of implementation. We are waiting for more people to enter the market and drive this cost down. And there currently is a huge requirement to be a specialist, to really be a bioengineer if you're capable of running these. And we really don't need that. We need turnkey automated systems that essentially, if you don't have any bench research experience, you could still run them within a clinic. So we need the technology to mature. We need to really robustly validate and benchmark tissue chips against animal models and against clinical data. And we need quality standards, which we're working on. And the image here really just shows you that if you are in a specific stakeholder group with tissue chips, you have different needs that need to be addressed, and they're not all overlapping needs. So we created an MPS World Summit that we funded for 3 years, as well as an international MPS society that came out of this to really try to train the next generation of scientists and meet and have discussions on standards and how we can improve the field. And you may ask, what's next for the program? We are now pushing into more of a regulatory space with our translational centers for microphysiological systems. And so these awards will be awarded in December, is our hope. Teams will actually take 4 to 5 validated tissue chips with very specific contexts of use. So for example, you could have a liver chip that you could use for tox testing, and you could take that same liver chip as a second context of use for disease modeling and efficacy testing. That's 1 each year, because this is a 5-year program. So it's not as robust as a timeline as ARPA-H, but it's still going to be more than we've ever pushed them to do. We'll be working hand in hand with the FDA, which we have since the inception of the program, and we find their oversight to be extremely critical. We're not going to be able to push this technology forward if we don't work hand in hand. And so really having a qualification pipeline and partnership, either through iStand or biomarker qualification program, there are a number of routes that we could do this, to eventually qualify these as bona fide drug development tools that can be used and accepted in IND applications in the future. And then the goal is to eventually scale that up, make those publicly and commercially available. And so I'm at about 20 minutes. And with that, I just really want to thank our consortium and everyone who's made this possible. I know it's really, it's a little lame to say at the top that partnership and collaboration equals program success, but our joke is that we really do play well with others at NCATS, and what we really want is cooperation and coordination. A lot of science is done in a bubble, and that's not going to help us succeed. I want to thank my program director, Dan Tagle, who is a completely innovative and wonderful person to work with, my program officer, Dimitri Krepke, and program analyst, Chris Sundrick. I also want to thank the FDA, EPA, our partners at BARDA, NASA, the VA, ISSNL, and CASIS, and our pharmaceutical partners that we meet with to kind of discuss the state of this within pharmaceutical development in the IQ Consortium. And last but not least, our Trans NIH MPS Working Group. We're about 60 program officers at this point, and we meet monthly to kind of discuss this program, ways to improve it, things that need to be, issues that need to be addressed, and new funding opportunities that we could put out for solicitation. So thank you so much, and thank you.
Good morning, everyone. It's very exciting to be here with you today to discuss the future of the NITRO program. There's some incredible opportunities that will come out of ARPA-H, and FDA is delighted to be here and hopefully help you on your way to regulatory success. My name is Amanda Suchik. I am a pharmacology and toxicology reviewer at FDA's Center for Biologics Evaluation and Research. That's CBER, the acronym that you'll see throughout my presentation, specifically within the Office of Therapeutic Products. Just to give you a little bit of background on myself, in case you're interested in asking specific questions later on in the day, I'm a classically trained comparative physiologist, and I'm also a veterinarian with expertise in laboratory animal medicine in species ranging from rodents to swine, small ruminants, and non-human primates. So without further ado, let's talk about the FDA and you. So just to give you a little bit of background on how I plan to structure the talk this morning, I'll talk to you a little bit about the organization of CBER and specifically OPT, again, the Office of Pharmacology and Toxicology, considerations for developing a non-clinical testing program specifically for CGT products. So this is cell and gene therapy. The focus of NITRO is regenerative medicine, so a lot of the therapies that you're developing will likely come through the Office of Therapeutic Products. some considerations for putting together your farm tox program, opportunities for early interaction with FDA in terms of formal meetings, your IND package, and then I'll provide you with links for specific agency guidance documents that will be a good reference point for you as you start to put together your regulatory packages. So CBER, again, FDA's Center for Biologics Evaluation and Research, has 3 different product review offices. So that's the Office of Vaccines Research and Review, the Office of Blood Research and Review, and then there's my particular office, the Office of Therapeutic Products, which was actually just reorganized earlier this year into a super office to allow the agency to expand that office in time as more cell and gene therapy products are being developed. One of the great things about being a reviewer with OTP is the wide variety of products that we regulate. So the technology that comes through our office is truly amazing and humbling. And we look at everything from gene therapies like adeno-associated viral vectors to various types of stem cells across the continuum of development, functionally mature and differentiated cells, products for xenotransplant, which has been in the news a lot lately, therapeutic cancer vaccines, blood and plasma-derived products, including anti-venom, anti-sera, tissue products, devices specifically for the administration of biologics. And there is a specification there because other types of devices would go through our Center for CDRH. And then combination products, which in this context is usually going to be one of these plus some type of a device. So the first guidance document that I want to point you to is this one. If you have to look at one OTP document that's going to kind of summarize how you put together an IND, it's going to be this one. So this is guidance for industry, preclinical assessment of investigational cellular and gene therapies. This was released by the agency in 2013. This is available to you on the FDA website, and I've also included a link at the end of my presentation so that you can find it easily. So this will give you an overview of what is expected from FDA in terms of your program design and what the objectives of your non-clinical development program should be. We'll give you specific recommendations for different categories of products. So again, cell and gene therapy versus a therapeutic vaccine. And then a topic that is near and dear to my heart, again, I am a laboratory animal veterinarian. FDA is very encouraging to sponsors to implement the 3 Rs when you're developing your non-clinical testing strategy. So for those who may not know, that's reduction of animal numbers, refinement of procedures to reduce discomfort, anxiety, and pain, as well as replacement of animals when that's a possibility. So getting to the meat of what you guys want to know is what does the agency expect in terms of your non-clinical testing strategy? So much to the frustration of, I'm sure, of all research teams is that there is no one-size-fits-all approach, specifically for cutting-edge new cell and gene therapy products. Your non-clinical testing strategy should be specifically designed for that product class based on the cumulative knowledge in the field of that class and then your experience with the technology that's available at the time, so the most current. It should be, you'll hear me say this many times, evidence-based and data-driven. So if you make a claim to FDA in sort of any one of your regulatory documents that this is the way that X works, we're always going to be asking you, where's the evidence from either your own original data or from the published literature to back that statement up? What I can tell you, though, is this. So when you're considering putting together your non-clinical development program, the data that you collect for an IND should consider these points. So the risks that are associated with a specific product class, the disease status of the target clinical population, the proposed delivery procedure and whether or not a novel device is required with that, the data from relevant in vitro and in vivo models to assess safety and biological activity, and then any other availability of existing therapies. So let me just give you a little bit of a scenario as to how let me go back one here. How the agency views your response to these questions. So if you're looking at a disease indication that is life-limiting with very little options for treatment, the FDA is probably going to have a higher tolerance for risk in that case than if you've got a non-life-limiting disease with several treatment options that are approved and available at the time. That's not saying that it's not worth submitting applications for new approaches. We always encourage that. But the agency's job is to look at the data that you submit and to make a judgment call as to whether or not risk is outweighed by benefit or the opposite direction. I'm sorry here, struggling a little bit. Okay, so for your non-clinical data, what should it accomplish? The first thing and the primary thing that I'll drive home multiple times this morning is that your non-clinical data should provide a comprehensive safety assessment of your clinical product that identifies both acute and chronic toxicities, as well as local and systemic toxicities following administration. You should provide your rationale for the administration of the product to treat a specific clinical indication. So this is your mechanism of action. And again, that should be supported either by original data or from data in the published literature. And then finally, your non-clinical testing strategy should provide recommendations to inform the clinical trial, specifically with respect to starting dose level, how high the dose should be pushed, and then the schedule at which the product should be administered. The route of administration that is used in the clinical trial should be reflected in the definitive safety and activity studies, and then the model that you choose, so the degree of disease severity, should reflect the eligibility criteria in the clinical trial, as well as the monitoring plan, specifically if any toxicities are found in your toxicology study. So, for the FDA, safety is always your first consideration. Activity is important. We're definitely not saying that's not the case. But at your level of development, so first in human clinical trial, safety is going to be the sort of benchmark that FDA is looking at first. This quote is from the Code of Federal Regulations. I'll let you read that on your own. Just kind of keep this as a maxim as we discuss this issue later this morning. So where can your non-clinical data come from? Well, there are several different sources, and they're listed here. It can come from appropriately designed, well-executed studies that you conduct in-house. It can come from GLP-compliant tox studies that you farm out to a CRO. It can come from published data in peer-reviewed journals. I do want to put a little bit of a caveat here. So, typically, peer-reviewed journal evidence will be for your activity or your proof of concept studies. It's uncommon that your safety data is going to come from this source. Also, with respect to activity, remember that for the definitive activity in tox studies, FDA is going to be asking you to specifically for data relating to your clinical product. So that means the specifics of the manufacturing process, the lots that you'll be administering clinically. So even if you have data that's published about a similar product, it's not your product. So this is helpful, but not necessarily to be used as a replacement for doing your own studies with your clinical product. Cross-referencing to similar products that have been filed with the agency previously, there are some rules involved with that, so make sure that you talk to FDA before you want to go that route. And then detailed clinical study reports from trials in people that were either conducted domestically or abroad. Generally, at this stage of development, like so IND, these are going to be small enrollment, compassionate use that might be like an investigator-initiated trial that will often be submitted by sponsors in addition to their non-clinical testing paradigm. So what everyone wants to know here at the NITRO OA Model Symposium is, what's the best animal? What should we be using that's going to get me the most purchase power with FDA? So, like a lot of people have already talked to you about, there is no necessarily one perfect model for all situations and all products. What I can tell you are the general principles of model selection. So, your model should mimic the target clinical population in anatomy, pathophysiology of the disease, and specifically for this crowd, I would emphasize biomechanics of joint loading. Your animal models should demonstrate a pharmacodynamic response to the clinical product. I think the best way to illustrate this is more with compounds that are regulated through CEDAR. So if you have a, let's say a small molecule that interacts with a specific receptor, and your animal model doesn't happen to have that receptor, it's a poor model choice for that particular compound. If you're looking at gene therapies, your model's cells should be permissive to the vector that you're using or the genome editor in the case of a CRISPR product. And the animal itself ideally should permit the use of a clinical delivery device. So where this would come into play, we see this with a lot of neurological products where the proof of concept studies may be done in rodents, but because, let's say, you've got a big halo system and a big catheter delivery system, you're not going to be able to use that in a rodent. So then you would move to a larger animal to ensure that you can incorporate the use of that device. Okay, so I'm going to use this photo just to illustrate the talking points on this slide. So models specifically for osteoarthritis. Always, you should be thinking for any disease process to use a comparative approach to your model selection. So what's the comparative anatomy of the skeleton in general, as well as the joint that you're specifically targeting? What does the pathogenesis of the disease look like in that species? So many animals will age at a much more rapid rate than a human being will. You need to consider that. And you need to consider the timing of when your clinical product is administered to that animal and the development of the disease in comparison to a human. So what you're looking at here, this is a fully anesthetized horse with its tarsus in an MRI. So why did I put this photo up here? To talk about these practicalities that you should consider before you start going down a road that you might not be able to handle for a variety of reasons. So when you're working with a human, let's say that you do a local block to, collect some type of a tissue sample from an extremity. In some cases, with a large equid, like a horse, you might be able to get that, but you might have to fully anesthetize that animal to get a joint tap or a sort of synovial biopsy, okay? Where are you going to do your in-life and your post-mortem assessments? So from someone, again, who's a veterinarian, it takes a really big room to necropsy a horse. Where are you going to house them? So if you're at a land-grant university, you're lucky in the sense that if they have a college of agriculture associated with them, you might have facility for equine. If you're at a vet school, that may be a possibility. But if you're at an institution that doesn't have farms, this is going to be really tough to do. What about the expertise? Do you need to contract out to an equine orthopedic surgeon to be able to get those samples? What about your anesthesia technique? MRIs that are capable for a horse. I can tell you on one project that I worked on with sheep, we actually used the human MRI that was at the medical school, and there was a huge amount of regulation, as there should be, to make that happen. So I'm not saying that it can't be done, but it takes a lot of forethought and planning to make this happen. And then finally, and don't discount this, is the expense. So it is much more expensive to manage a research herd of horses than it is to manage 50 C57 black 6 mice. So just some things to keep in mind as you're looking for the ideal animal model. Ideal is a matter of perspective. You have to actually be able to get the work done and submit that data to the agency to get to the next phase of nitro development. So just keep that in mind. Okay, other considerations. This is just basic science, like rules of science, nothing kind of new here. Make sure that you have appropriate enrollment in your experimental groups. Do a power analysis. Enroll both sexes. Dose levels. I do want to put some time here, and I will come back to this at the end. So the dose levels that you choose for your non-clinical experiment inform the design of the clinical trial. So with respect to biological activity, the lowest clinical dose level, you should have non-clinical data that shows activity at that dose level. With respect to toxicity and safety, you should be going higher in the non-clinical data than in the clinical trial to give FDA a safety factor to see where, if there are any safety findings, how much wiggle room do you have before they start to manifest. For the major assessments that we're going to be looking for, that's biological activity, which we've talked about, safety, which I'll talk about a little bit more. I do want to focus some discussion here on distribution. Some folks tend to forget this one, and it's really important for the interpretation of the safety data. So if you have safety findings in your tox study, it's important to FDA to know whether or not those findings correlate with where the product went in the body or not. Distribution data can also give FDA a little bit of a heads up of toxicities that you may not have considered. This is the most common for distribution to the reproductive organs in both females and males. So if you're getting a lot of distribution to the testes or the ovaries, FDA might come back to you at the time of, at IND or even before that and say, hey, we need to be thinking about developmental and reproductive tox studies here, which are very expensive and very time and labor intensive. So again, something that you want to consider fairly early on. The dose level extrapolation. This is a big mistake that kind of rookie sponsors will make when submitting to the agency. You want to make sure that you lay out for FDA how you're translating a dose from a horse or a goat to a human being. Is the administration local? Are you basing your dose level extrapolation on surface area, on allometric scaling of body mass, on a variety of different dose level units? This extrapolation, as a reviewer, I should not have to work hard to make that leap for myself. You should show me this in a very logical and evidence-based way with supporting data. The clinical testing paradigm, we talked about that, how the non-clinical dose levels should match. Device compatibility, this is another sort of rookie mistake that some sponsors can make. I'm going to use cells as an example. So if you're administering cells through a device and a lot of those cells get stuck in the device and they don't actually make it to the animal model or to the patient, FDA needs to know that the data that you're giving us in the tox study, is that actually reflective of what you're saying the dose level is? Or did they actually get less than that because a lot of the cells got stuck in the device, and that the toxicity is actually occurring at a lower dose level than what you report? Biocompatibility, carcinogenicity, and tumorigenicity are specific cases that will depend upon the product. Again, a good example here would be a cell product that is seated on some sort of a matrix. The matrix may be considered a device, in which cases biocomp studies would be required. If you're giving some kind of an iPSC product that requires transformation, FDA is going to want to know if that transformation isn't 100%, is there a risk of neoplasia in your recipient? Okay. So the take-home message, your non-clinical development program should be evidence-based. It should enable FDA's assessment of the benefit-risk ratio. It should be designed with the best available technology that exists at the time of your submission, incorporate judicious use of animals and their numbers, and then inform the clinical trial with the data that you've collected. All right, let's talk a little bit about how FDA can help you early on in your program. The first opportunity for formal interaction with the agency is called the Interact meeting. This will happen during your sort of discovery development phases. There's a link here so that you can learn more about it. You can look at it at your leisure. I'm just going to talk about what the goal is. So Interact stands for Initial Targeted Engagement for Regulatory Advice on both CBER and CDER products. The goal for you is to obtain early feedback from the agency on product development. These are non-binding discussions between the center and the sponsor, and the goal is to discuss specific issues. So what you're not going to do at this stage is be like, I'm going to submit my entire preclinical development program to FDA and say, what do you think, FDA? Instead, what's going to happen is you're going to say, FDA, can you give me some advice on my dose level extrapolation? How do you feel about, FDA, about the biomarkers that we're using to assess activity? So again, targeted engagement. And the timing, again, is when you've generated preliminary preclinical data, so proof of concept and some safety, but you're not yet ready to conduct definitive non-clinical studies. One caveat that I want to put on Interact meetings is that these are granted at the sort of discretion of the agency. So you submit an application for an Interact, and then FDA will come back and say whether or not that's granted. Interact meetings are generally granted for cutting-edge technologies with which the agency doesn't even have a ton of experience with. Next one is pre-IND. So this one is one-to-one. Everybody gets one pre-IND before they submit an IND. And this is sort of happening in the middle of your non-clinical development phase. So the goal, we want to be able to give you feedback that allows you to achieve a successful IND application. These are non-binding scientific discussions between the core review team, so that's chemistry, manufacturing, and controls. That's me and Pharm Tox, as well as clinical, to comprehensively communicate you to us your non-clinical development program, and then discuss, are there any deficiencies in that program, and how can we get those rectified before you submit your IND? Timing, after the POC and preliminary safety, but before the definitive safety study. Okay, and then finally is your submission of your IND. So every product that's novel, that's going into a person and is being sort of investigated as a clinical trial and is a drug or a biologic requires an IND. IND should have all of the necessary information regarding CMC, Pharm Tox, and then clinical data. IND and the specifics of it is a little beyond the scope of this particular meeting. I think that pre-submission discussions are much more germane for sort of where NITRO is at this point. So I don't have a ton of detail here, but I'm happy to talk to you about it in small groups later on. If you have specifics at any one of these stages, we can talk about it. So to summarize, OTP is within CBER, and we regulate a whole bunch of different products that are germane to sort of this audience. The non-clinical development program for any particular product is unique to that product, and there's not a one-size-fits-all. The non-clinical data should demonstrate the safety and the activity of that product at the dose levels that you intend to administer in the clinic. You should understand the limitations of your test system, be that in vitro, in vivo, or in silico, and you should understand the benefit-risk profile of your product and then communicate that to the agency. And then don't forget, we're here to help you. There are opportunities to obtain FDA feedback on your non-clinical development program at multiple steps along the way. So here is a list of guidance documents that I think would be helpful to you. I think this third one, again, our general non-clinical assessment of CGT products is a really good place to start, but there are others here that are relevant as well. These are available on the FDA website or through these links that you can get through a copy of my presentation. And then here is my contact information and just some associated Sorry about that. Links in case they're of interest to you. My name is Amanda. Please feel free to contact me if I can help you with anything specific. And on behalf of FDA, I'd like to say we welcome your submissions and we wish you every success in your development. So thanks very much.
Okay, good morning, everybody. So I'm going to be a small part of the talk today from NASA and ISSNL. Mike Roberts will give you most of the information about things to do with tissue chips and things that have flown in space, but I want to talk to you a bit about biological and physical sciences, which is also getting involved in the tissue chip initiative. So just to tell you about the problems that we have in space and the things that we have to consider when flying in space and how this affects our biological systems. We have, of course, microgravity, which in relation to muscle and bone, there is unloading. Okay, so we're getting a great deal of unloading within the situation. Then we've also got the ionizing radiation effect. Now, when we're on Earth, we're protected by space radiation. This is going to sound a bit more like sci-fi, right? With galactic cosmic rays and solar particle events. But our magnetosphere around the Earth protects us from these getting to the Earth, or most of it. Whereas low Earth orbit will definitely get more of this radiation. And as we start thinking about going out beyond LEO and going to the moon and going to Mars, then this is going to become more of a concern. There is also effects on the human body in terms of daylight cycles, in terms of we're in a closed environment, pressures will be different, carbon dioxide will be different, which of course can alter our metabolism. And then isolation, and there's things like regolith, and when we start to think about the moon. But when it comes to this audience, effects on bones and muscles, we've really seen some effects when it comes to the radiation, the space radiation, and the microgravity environment. So space biology is more to the left of the slide. We are involved in basic fundamental mechanisms and understanding how the biological systems change. We use all sorts of different types of models, our highest being, I suppose, the rodent, right? But we have the cell culture systems, the tissues on a chip, but we have invertebrate models too, things like worms, things like fruit flies, and understanding also how things like plants will grow in space and microbes and how they interact with the microbiome in the human setting or even with plants. So now when we come to more the translational side, we actually have, the animal research, which, as I said, probably the highest we have are the mice and the rats. We have effects that we're seeing there's an altered immune system. When you go to space, we lose bone, we get muscle wasting, we have alterations in connective tissue. So there's lots of different things going on in space in humans, and we also see these in our rodent models. Now, as well as the effect of microgravity and the unloading of, the unloading on the muscles and the bones, leading to the bone loss or involved in the bone loss and the wasting, we've actually seen experiments on the ground done with space radiation, which we have a National Space Radiation Lab at Brookhaven National Labs in New York State, which can simulate GCR, galactic cosmic rays, by using a number of different high-energy particle radiations, as well as protons. And what we do see is that will actually alter osteoclast activity and, in fact, differentiation. We see much more of the osteoclast differentiation, and so it may be that the radiation that we're exposed to in space is actually contributing to bone loss as well as the unloading. Now, so the issue with experiments in space, one, they're expensive, as we know, and they can be very complex when you're dealing with a rodent. When we send rodents to space, we're talking about there are 10 mice in a habitat, less if you use a rat. and we may have 4 habitat potentials to go up. Incredibly expensive, a lot of astronaut time involved in maintaining those animals and doing the experiments. So an alternative, of course, is the tissue chip. So that's where we come to today, is the use of the tissue chip to look at the effects that we see in space. And we're hoping that these are going to be able to be applied and move further so that we as BPS, Biological and Physical Sciences, work on fundamental mechanisms that then will transfer over to the human research program so that they can determine their risks, so that they can determine maybe countermeasures. And again, the tissue chip would be a way of checking on this and to be able to use this. So, you know, as we've been talking about, there's bone on a chip, we've got all sorts of different types of chips now available. We're looking at understanding mechanisms of what's happening in spaceflight, evaluating and screening for adverse health effects with pharmaceuticals, and also the importance of this, we can increase our N number, right? We can also increase our cell population type that we're looking at. We're not necessarily just looking at an inbred strain of mouse now. We can actually use cells from people with the adult-induced pluripotent stem cells. So that this is a way of bridging, right? So we've got a lot of information that we found out about rodents, but we really want to pursue further, understand much more about mechanism. So we're hoping that the tissue on a chips is going to do this. And of course, there are all sorts of tissue on a chips that have been generated, as well as organoids. And again, the organoid allows you that help in that you can get a cell village, a cell population. You can take a number of induced pluripotent stem cells from different lines, mix them, differentiate them, and you end up with like a cell population. Now, so reasons for going to space. Why would you send your experiments to space? One of them is, as I've been talking about, to understand the mechanistic issues to do with the stresses of space, the combinations, you know, the radiation as well as the microgravity. However, NASA is also very interested in developing things in space or experiments in space that are going to benefit humans on Earth. One thing that we're finding, whether it be in the humans, the rodents, or the cell studies, is that space seems to have almost an accelerated aging phenotype, is where it's potentially looking at. We see mitochondrial dysfunction, we see oxidative stress. And so there is that reason to consider about your experiments in space. And so there was Lisa Carnell, who's our director now of Biological Physical Sciences, and in who I'm standing in for today. We have been working with multiple agencies, and there was in 2022, 9 awards, $18 million put into with the different agencies to develop tissue on a chip. What thing's going to be important is automation and extensive time in culture. So like 6 months, we want to be able to get those chips out to 6 months. And in fact, this was just one of the studies where they have bone marrow with osteoclasts and osteoblasts. So this could tell us something in space about what's actually happening with those cells. And also about the immune system, which is, of course, important in a disease such as osteoarthritis. And then, of course, we have Avatar, which is going to be on Artemis 3. So this is going to actually be 2025. It's going to sit on the surface of the moon. This will be a tissue chip, and it will be in a self-contained unit that will maintain the whole chip for the extent of the experiment. And so this is where we're hoping to go. And thank you very much. And I'll hand over to Mike.
So to pick up where Lynn got us, so we have access to space to further human exploration goals. So NASA, of course, as everyone in this audience will be familiar with, has been at it for a long time. And human space exploration has always been a very prominent part of that portfolio. With the building and launching of the International Space Station, NASA also began to look at access to space as enabling discovery, research and technology development, and even translational applications in space that not only benefited NASA's goals supporting human research, but began to look at space as an opportunity, a novel environment in which to accelerate the pace of biomedical discovery and technology advancement that would have direct benefits here on Earth. So over the 22 years that we've had the International Space Station and operating in orbit, we have benefited here on Earth from that continuous human presence in space, in that a multitude of different scientific areas have been advanced, many of those supported and funded by NASA and its programs, but also more importantly and continuing to grow more recently are the involvement of other US government agencies and international partners in supporting research that may not necessarily directly benefit the NASA exploration goals, but are important to the missions of each of those agencies. The ISS National Laboratory came into being about the same time as the National Center for Advancing Translational Sciences at NIH, independently, with the goal of having access or increasing and enabling additional access to the International Space Station for the support of research for academic research communities, commercial, for-profit, and not-for-profit commercial entities, as well as to encourage partnerships, public-private partnerships, that could benefit from access to space. The International Space Station National Laboratory itself was created in 2005 by the US Congress in the NASA Public Funding Act of 2005. NASA operated the International Space Station National Laboratory for the first 6 years of its existence. And in 2011, there was a cooperative agreement notice that sought to have a not-for-profit, non-government organization begin to operate the International Space Station National Laboratory, with the goal to be able to engage with the private sector in ways that were difficult for NASA to do as a government agency. That solicitation resulted in an award to the Center for the Advancement of Science and Space, where I work. So I don't work for NASA, I work for a not-for-profit, non-government organization that has managed the International Space Station National Laboratory since 2011. As Lynn touched on, there are a wide variety of physical changes in the absence of gravity in that environment that have direct impacts upon physical systems that affect both energy and material transfer. Those also translate very rapidly into biological effects for biological organisms, whether they are at the level of tissues or cells or whole organisms such as the astronauts who fly to the International Space Station. So while Lynn spoke about biological physical sciences and human research programs at NASA, which are focused on understanding the fundamental phenomena underlying those and their impacts upon physical and biological systems, HRP, as she indicated, focuses on understanding risks to humans in that space environment and mitigating those risks through time. The portfolio that is represented at the ISS National Lab is more focused on the needs of other US government agencies or industry. So at the outset, we're not interested specifically in addressing the needs of crew and human exploration, but we are interested in access and enabling access to space to further humanity's goals here on Earth. So with that in mind, we've had active partnerships not only with NIH NCATS that I'll speak to in a moment, but also with the National Science Foundation. So we've partnered with the National Science Foundation now for 6 annual solicitations since 2018. We now have over 20 projects. I haven't updated this chart in the past few months, and well over $8 million in funding. This particular program is focused on those fundamental aspects of the interaction between the biological components and the system in which they're housed. So most of that work has focused on microphysiological systems and tissue chips, understanding the interaction between the physics induced by those systems and the biology. But more recently, it's starting to expand into the use of organ-based models such as organoids. That program has also done some fundamental research looking at the advantages of bioprinting, of 3D additive printing facilities on board the International Space Station and microgravity. So believe it or not, there are actually some real world advantages that negate all of the significant expense in getting materials and supplies to the International Space Station and low Earth orbit that actually demonstrate that there are some unique printing advantages to working in that space environment. And only in the past 3 months, a commercial company returned the first biological product from space for investigation, a knee meniscus, which is currently under investigation in their labs. In addition to those collaborations with other US government agencies on the fundamental end of the spectrum, NASA is also partnering with other institutions, and ISS National Lab is working with a variety of commercial entities as well in partnership with NASA to facilitate in-space biomanufacturing. INSPA is an acronym for in-space production applications. So NASA is interested in working with the Department of Commerce and other US government agencies and industry to expand manufacturing capabilities in low Earth orbit for the future. This, of course, is going to be required if we do in fact discover phenomena from the space environment that leads us to be able to produce superior biological products or even non-biological products in the space environment, so that we understand what the regulatory regime will look like and how to close the business cases so that even if you have a very significant increase in the value of a product or its utility for clinical application, that we can continue to drive down the cost of its manufacture using that environment. So what I've shown here is one example from a company that was a startup only a short 5 years ago, Lamb Division, which is seeking to leverage the absence of microgravity to improve the manufacture of by thin layer deposition of a replacement lens to combat macular degeneration. So through multiple flights now, Lamb Division has now flown 6 experiments to the space station over the past 3 years. They've been able to demonstrate a far superior product in their thin layer deposition of bacteria rhodopsin, which they believe will lead to faster clinical approval of their commercial product in the end. At the very least, they are learning from their thin layer deposition in the microgravity environment ways that they may be able to improve their manufacturing process here on Earth. So what we gain from exploring not only fundamental research, but manufacturing outcomes in the space environment does not necessarily mean that it has to translate into closing the business case for manufacturing items in space. We're actually starting to learn about the fundamental processes of those manufacturing processes that have direct benefits. In 2016, beginning in 2016, the ISS National Laboratory reached out to the National Center for Advancing Translational Sciences to essentially learn more about the tissue chips program and ways that we saw potential advantages to making as one component of the NCATS investment in tissue chips even more beneficial by offering them the opportunity to take some of those technologies to space. So that led to a partnership between the ISS National Laboratory and NIH, with NCATS issuing a solicitation that offered opportunities to take tissue chips to space. The reason that that was attractive to the National Institutes of Health is that exposure of these model systems to the space environment offers as an accelerator for the onset and progression of some human diseases. So Lynn spoke to, we have seen biomarkers and indicators that prolonged exposure to the microgravity environment and the stresses there, perhaps due in part to radiation, but certainly in major effect due to the mechanical unloading that affects bones, muscles, tissues, cells, leads to accelerated aging processes. That's not to say that you become older when you live in space, but that there are some effects on your body, at least, that clearly replicate what we see here in the natural aging process on Earth. So those accelerated models based on tissue chips offers investigators advantages in the utility of their in vitro models here on Earth. The NIH, NCATS, ISS National Lab partnership led to the selection of 9 investigator teams who were each offered 2 flight opportunities to the International Space Station in quick succession. Quick is a relative term. Quick for space here meant that they had about 24 to 36 months between their flight opportunities. So it takes typically 24 months to 36 months to ramp up a cell science-based experiment in a space environment, and then being able to turn that around and fly it again within another 12 months presented some severe engineering challenges for the community of professionals who engineer and design those chip systems. But it worked. So we are continuing to see results in peer-reviewed publications starting to come out of this partnership. So the way that NCATS approaches this technology development and discovery is similar to what ARPA-H does, in that there are selection gates as you move down the process. Those are very beneficial to work in a space environment because you have to have a very close collaboration between the engineering teams and the biologists and other folks behind the design and operation of the experiment. But we've already seen success in understanding the way that stem cell-derived heart cells perform in the microgravity environment. We've seen a continuation of that experiment where recently that flew as a part of a private astronaut mission. So in addition to having access to the crew on board the International Space Station to execute your experiment, there are now commercial companies that are contracting the services of their private astronauts to conduct research on station. The exposure time to microgravity there is more limited. This experiment went over the course of about 12 days, but they offer opportunities for even quicker access to the space environment. And what we've seen are folks starting to think about ways to expand the potential for manufacturing stem cells in the space environment so that you result in higher density yields and more homogeneous populations of iPSCs in a shorter amount of time. We've also seen significant progress in the design of the microphysiological systems for use in space. I call your attention here to a recent publication from Siobhan Mullaney's lab at University of Florida, which focused on using a platform to model age-related muscle wasting in microgravity. We also have, although I didn't include an example of it here, a microphysiological system that was flown with support from the NCATS partnership that was focused specifically on bone, cartilage, and synovial fluid from 2 ages, both male, in the space environment. So there has already been a significant amount of learning to benefit this community focused on the development of in vitro OA models moving forward. I wanted to include this chart just to get your minds wrapped around where we see the International Space Station and these future commercial platforms that will replace it in short order, offering an advantage not only to research and technology development, but also manufacturing applications and clinical applications here. So as I mentioned, the ISS National Laboratory was created to enable a pathway to low Earth orbit for the support of research that benefits humanity here on Earth. So the way that the ISS National Laboratory currently operates is if an investigator comes to us with financial support for their research, they don't also have to identify financial support for the cost of the rocket and access to the crew time. So as a rough rule of thumb estimate, each flight experiment that we do that involves biology is typically on the order of $7 to $10 million of US federal investment to conduct it in space. That resource allocation is provided to US research entities at no cost. So the intention here is for NASA to continue to expand the utilization of space, but in addition to enabling its exploration goals here again, the focus is on providing access to space for research, technology development, especially in the biomedical arena, that can be benefited and accelerated by exposure to that environment. In the very near future, the International Space Station is going to be retired. It's going to return to Earth and hopefully will land somewhere near Point Nemo in the ocean. But it will not result in a gap between the capabilities that we currently have available to us on board the International Space Station and what we see in the future. So at the earliest right now, barring some catastrophe, the International Space Station will continue to operate in space until the end of calendar year 2031, or maybe slightly beyond that. Commercial LEO destinations, LEO being an acronym for low Earth orbit, are currently in design and in some cases in assembly to begin operations across time between 2026 and 2030, with the goal being that at some point before 2030, there are at least 2 or possibly more commercial platforms operating in space that can fulfill some of the same capabilities that we currently have available to us on the International Space Station. So this is what the historical view looks like. The International Space Station began assembly in 2000. It took about 10 years to get through the decade of assembly, although science and technology development was ongoing throughout that whole time. But I'm sure each of you can imagine trying to conduct research in your laboratory while you're having it built and assembled. It was difficult at the time. From 2010 to 2020, we continued to revitalize the availability of systems on board the International Space Station and work out the logistics for doing complex, complicated experiments such as that is required for in vitro cell culture in space environment, as well as the rodent research program that Lynn mentioned. So beginning around 2020, we actually entered the first decade of results. So since 2020, we've seen a significant increase in the number of publications and science return from the International Space Station, in part, or in primary result of the investment that NASA and US taxpayers made in building the International Space Station. And I think just as importantly, in opening it up not only to NASA-funded investigators, but opening it up to researchers who were funded by commercial companies, by for-profit institutions, and even more importantly, by other US government agencies such as the National Science Foundation and the National Institutes of Health. Coming to space near you very soon are a multitude of commercial LEO destination platforms. So these are the laboratories of the future. What I've shown here are a few of the consortia of companies that are currently designing and in some cases actually beginning to build their laboratories of the future. Axiom Station will begin life attached to the International Space Station beginning around 2026. And over time will build out in modular form until it establishes its own power regeneration system, and then will become a free flyer platform that separates from the International Space Station. Orbital Reef is a consortium between Blue Origin and Sierra Space, which plans to begin life as a free-flying crewed environment similar to the International Space Station itself. Starlab is a single launch vehicle, primarily based on an inflatable capability that will offer single launch capabilities that can either be crewed or uncrewed. And Northrop Grumman Space Station is looking at taking some of the design criteria that they've put into operating and being part of the Artemis moon approach into developing a system that could operate not only around the moon, but also could operate in low Earth orbit around the Earth. And as happens, there are also a multitude of companies from around the world who are beginning to build and operate systems that did not directly benefit from access to NASA funding. So Varda and VAST are what I like to colloquially call the pirates that are working in space now. The Varda system is designed to be a crewed habitat, which will be launching and operating in low Earth orbit sometime before 2028. And Varda is already on orbit and has manufactured what they believe to be an improved crystalline form of a frontline therapeutic agent in space and is currently waiting for FAA approval to bring it back home. So this is not science fiction, it's now science fact that we have access to low Earth orbit and space for not only advancing biomedical discovery and translational outcomes, but folks are actually starting to get funding from VC world, from venture capital world, to support manufacturing in space. With that, I'm done. I will be here this afternoon. I think Lynn has to depart this afternoon, but I'll be glad to take notes on questions so I can get them to her so she can address them from the NASA side. Thank you.
Can we get another round of applause for all of those speeches? Those were fantastic. So thank you to Gail, Pasley, Amanda, Lynn, and Mike for those amazing speeches. You guys had a very cool and novel snapshot. Thank you for also being here. Your input is going to be critical to define what the future looks like for this entire industry, sub-industry, and the path forward, apparently on Earth and in space. We always like to end with a slide for the agency. This is over at the Renwick Gallery, not too far from here. The present moment used to be an unimaginable future. Obviously, that's true when we're talking about science fiction now being scientific fact. As a kid growing up, I would come to the Air and Space Museum all the time. My family's from here. I'm actually a descendant of the Mattaponi tribe. So to be able to have ISS speaking and NASA speaking and be right next door and be on this stage with the Smithsonian logo is a very humbling and surreal experience. So thank you. What we're about to do for Nitro and the future we're about to create will change everyone's mind so that in a decade when they look back, they'll say, you did TKAs every 10 years? So I think that would be a good time to break. Again, thank you to the speakers. Thank you for being here. And we'll see you this afternoon. Thank you.